Hi. We are back. Ooch. We'll have to click through some of these because I don't want to like go over all the questions again. We'll get everybody joined in first. All right, so we'll go over the litmus stuff just because I have the time to do it right now. Let's get signed in. All right, with litmus, we know our neutral starts out as lavender, right? You already said that over there. We're familiar with what the milk looks like and all of that. So here's our neutral. This is our starting point. All right, if you're making acid, when you're fermenting, what sugar do we ferment in the litmus milk? Do you guys remember? Lactose. Yes. So fermenting lactose. You'll make acid as a product. Acid, we know that's going to be pink. That's your first stop. Okay. If you're pretty aggressive with this, then this one will have reduction of litmus. This one is white. If we reduce litmus, Leo says GR, right? We're gaining an electron. If we're gaining an electron, we're no longer really litmus anymore. We're litmus with an electron. We don't work as litmus anymore, and therefore everything about litmus is lost. We're not purple, we're not blue, and we're not pink anymore. We're just not. So that's why we get the white. The white is representative of the milk color. Okay, so that's from aggressive lactose use in that case. So we're starting to see how that lines up with EMB. We also see how it lines up with McConkey. We also see how it lines up with the PR fermentation tube for lactose fermentation. And we have the TSIA that has lactose, sucrose, glucose. All of those have lactose fermentation, including litmus. So next we have, that's our acid side. This is our fermentation side. Both the P and the W guys can coagulate. So this is solidification or curdling. Okay, that can happen with any of that acid side of things. On the base side, over here, we are breaking down the protein casein. It's a protein, it's just a protein that's in milk. Break it down or use it, and we make an alkaline product. This one is blue. So that's our first stop there. If you completely break down all the proteins, then this one, we start seeing transparency. Let me do this. Which we call this digestion, because we completely digested all the proteins. And then this one is transparent. it looks like you can see through the tube like it's plasma, and you'll have like that layer of kind of purplish at the top of it. So that's because the litmus is still there. It's still purple. Purplish blue, but it's still purple. It's still there. It hasn't been reduced, right? But it separates out because that digestion is happening here. Okay, so that is our litmus chart, if you will. Also, if you have coagulation, we could have gas. That is a possible result that we didn't have any tubes that did that, but it is a possible thing. You would see bubbles or cracks in the media if that was the case. I like this better than the charts, like the tables, I mean. That helps me visualize what's actually happening. All right, so let's get going. We'll have to skip through the first few questions. That's all right. I think we'll survive. We're still going to finish way early, so. Next. The problem is I have to wait for the thing to pop up to skip it. That's the problem. I can't like select a question. So we get to do this until we find citrate again, guys, so bear with me. That's the only thing that sucks about the Kahoot stuff. If you guys have any questions about these while we click through them. First step in the gram stain. We know that's, we're still a good try, guys. It's like, now it's like, I know. Now it's like, if you can beat my click clock on news. Super quick answer. I need to get rid of this too if you guys are gonna. This one was and it's spore stain. This one was, the problem was the plates, man. I really wish I could just select the question. Sorry. Beat the bullet, guys. Heat fixed. Where do we use heat again in the acid fast? No. Unless you guys have a little bit of joy in that for a second. We were pretty far in. I didn't realize how far we were into it.
Yeah, we're like halfway to the 20.
All right. The primary stain and the capsule stain. Congo. All right. Staining procedure that does use steam and a spore. It's kind of mini review of the review. All right. The media that was here, the first one, I believe, just from memory. EMB. All right, remember that's that metallic green that gives it away. What is the selective agent for EMB? EMB. All right, which media is this one? This one was McConkey. And then we're almost there. This one was McConkey selective or differential. Which ones? It was both, exactly. I'll give a little bit of joy to some of you guys. This one takes a little longer. All right, calling it. Good job. All right, which media was this one? That was mannitol salts. All right. pH indicator for mannitol salts. Red. And I'll put the screen down once we get to the right thing so we can see this better. Although I'm not sure it really matters, but. All right, here we go. Now we're getting close. So that was J, that's right. So I'll let you guys get this one. Now we're back on track. Citrate, no, citrate, green. Citrate and malonate are citrate is the plant, malonate is the broth, right? We'll start green. And then they both can turn blue if they make the alkaline byproduct from using that carbon source as the carbon source. So which of those tubes was positive? We know that one was. The blue means you have an alkaline byproduct. All of these always start out as neutral. They don't become neutral, right? And outline means blue, or blue means base, outline. And now you know this one would help to do that. which one's the positive one, which one's the neutral one. Because I might just put a blue tube and a green tube and be like, which one's the positive one? All right, what is happening to that pH in tube B? We went over this just a second ago. Let's see if you can figure this one out. All right, so this is a matter of understanding the pH scale. Remember, pH goes from 0 to 14, 7 being neutral. 0 is the acid side, 14 is the base. So that's just a refresher on that. In order to know that, if we're at alkaline or base, we go up from neutral. Right, know how Simmons citrate is inoculated. Simmons citrate is a slant, and we inoculate like a regular slant, right? Surface slant. How do we read it? We're looking for the color change from green or blue, colors that are produced. We start out green, we might turn blue. What does the blue mean? We made an alkaline product from using citrate as our sole carbon source. And what is our pH indicator? Bromothymol blue. All right. Which test is this one? Okay, so this is the phenol red fermentation test. We have two tests that we do that have Durham tubes in them. That's what that upside down tube is called. It collects gas from whatever the process is that we're testing. In this case, this is phenol red carbohydrate fermentation. It has a Durham tube. So we know if it has a Durham tube, it has to be either carbohydrate fermentation or nitrate reduction. Those are the only ones that have Durham tubes. And this one has color. It was that coral red color. So we know that it has to be phenol red carbohydrate fermentation. That's how we'll identify that one. What is that name of that upside down tube? All right, so that's the Durham tube. Collects the gas that's made as a product of fermentation. This is kind of part of the next question, relates back to that. So what are the two products of fermentation that can be detected by this PR, carbohydrate fermentation test? Two products by this specific test. All right, so we can obviously test for gas. We've got that Durham tube in there, but we're also testing for acid in this. So that's our other fermentation product. You ferment, you make gas, potentially. But if you're fermenting, in our case, for our purposes, you will make acid. That's always for fermentation. Sometimes you make gas. All right, which test is the one that's pictured here? This is one test. Take a minute to look at the tubes. The two on the right have stuff at the top of it. We're going to talk about it. All right, so this is the SIM test, sulfur indole motility. I think it's going to ask about I'm pretty sure. Yeah. What's it stand for in SIM? All right, so that's indole. We're going to talk about TSIA in a little bit, of course, but what does the I for TSIA stand for? Do you guys remember? Iron, right. And so does it have iron in the name? Do you guys know what it's for? So yes, the iron is like what's in the media to detect sulfur reduction. So yeah, lucky we have choices for you guys. But yes, so they both test for sulfur reduction. So don't forget about that. Just wanted to bring that up. Oh, that's up there. So how is indole detected in SIM? Our indole is detected in STEM by using Kovacs reagent. So that's what that layer was at the top of the tubes on the right for those 3 tubes. When Kovacs reagent turns red, that meant that indole is there. That indicates that your organism broke down tryptophan into indole. Otherwise, it stays that golden color. So the gold is negative. Okay. How do you detect H2S or dihydrogen sulfide in SIM? All right, so this is going to be observing that black precipitate, which we saw in that 3rd tube in the example it gave us. All right, how is motility detected in the SIM test? If we look at these pictures here as well, do these tubes look like they have modal or non-modal organisms? Modal, yeah, we don't see that clear defined stab line that we usually see with not. So we have growth away from those stab lines. It's either going to be foggy in the media, or you'll actually see the growth away from the stab line, or you might not see the stab line anymore, but there's clearly bacteria. How do you inoculate SIM? This one's a butt. We're going to stab it. How do we interpret the results? For the sulfur part, we're looking for a black precipitate. If there's no black precipitate, it's negative. For the indole part, we add Kovacs reagent. If it stays golden color, it is negative. If it turns red, it is positive. For motility, again, growth away from the stab line. The only reagent we use here is Kovacs. Which test is the one that's pictured here? That's litmus. It's got that nice lavender color. There's a lot going on with litmus, so I'm hoping that that chart that I made is partially covered, but I guess we're following it well enough. In litmus milk, what color is associated with an increase in the pH for your pH scale? All right, so an increase in pH is base or alkaline, and that's going to be associated with the blue color. In litmus milk, white, what does it mean? All right, so white is reduction for this one. You can have white color and not have coagulation, so I'll let you guys know that too. But if you have coagulation, it can be white or pink. All right, so our interpretation of our colors, we kind of went over that in our little chart over here. What is happening with digestion? If you have digestion, it's because you have completely broken down the proteins in the milk. How is gas detected? We'll be looking for bubbles or cracks in a coagulated medium. And what is the sugar in milk? Lactose, right? Or which test is this one that is pictured? So this is not the media color itself. We've added something and it's made it change this color. It's dark color. All right, so this is the starch hydrolysis test. We're gonna go over it. All right, which organism that is pictured in starch hydrolysis is positive for starch hydrolysis? All right, so that was the left organism. We see that halo where there's not the dark color. That's because there's no starch there to interact with the reagent that we have to add. The reagent interacts with the starch and causes it to turn that purple brown color. What reagent is that? That's gonna be iodine. All right, which test is this one? So notice that there is a Durham tube in there. That in there. Didn't I?
No, you had it on you.
Oh, did I?
Yeah.
Oh, it's right here. Weird place, but it's here. Weird when like those things hit you and all you're like, oh my gosh, what do I do with the thing? Okay. So that's nitrate reduction. Remember, two tests that have Durham tubes. Carbohydrate fermentation, which is that orange color. Nitrate, which is that kind of neutral, not color. What do reagents A and B detect in that nitrate reduction test? A and B. We only really test for two things in this test. The first one, A and B, is looking for nitrite, right? A and B, first in the alphabet, that's the first step. Last in the alphabet, late in the alphabet, zinc, late, nitrate. So the other one's nitrite. So if you have nitrite, you'll turn it red with A and B. You're positive for nitrite, you are therefore positive for the nitrate reduction. I know. What does zinc detect? So we got A and B out of the way. What does the zinc detect then? If zinc comes late, it tests for nitrate, okay? Late in the alphabet, late in the test, test for nitrate. Our little mini test just with the zinc, we're just looking at only that one. So taking our product that had the A and B in it, didn't change the color, we add the zinc to it. Now, that's our mini test. If it turns red there, we are positive for nitrate. So our overall nitrate reduction test, did we reduce nitrate overall? Nope, because nitrate's still there. Good job. Believe me, you guys are getting this way better than most semesters do. So, all right. So nitrate, know what we detected each step. Nitrite, the first A and B, nitrate, the second was zinc. Red is not always positive for the test overall, right? So be sure you understand the mini steps. And I feel like then you can go back and ask, did you reduce nitrate? And don't forget, you could make gas in this. None of our tubes did, so we didn't have an example. If you did make gas, you're changing nitrate. You have to make the gas. So that would have to be positive for nitrate reduction if we saw gas. All right, which test is the one that's pictured here? This is all one test. I know you guys feel the pain. Electrons. Power save for some reason, nobody knows why. All right, TSIA, triple sugar iron assay. You guys got the iron. I'm very proud of you. Do you guys remember what the three sugars are? Lactose, sucrose, glucose. Good job. All right, you can choose more than one for this. Which of the tubes that were shown show evidence of dihydrogen sulfide production? So we're talking about sulfur reduction here. Sulfur got reduced to H2S. We gained the electron. So we made H2S as the product. When oxygen gains its electron, it makes H2O as the product. All right, so this one, we had the 3 and the 5, the ones with the black precipitate. All righty, so still black precipitate for both of those tests. In the TSIA tubes that were shown, which ones show evidence of gas production that doesn't involve H2S? Just itchy, the stress itches me. Okay, good job. So this is gonna be the 4 and 4A. You can see the auger actually is like lifting up out of the tube. That's from the gas production from fermenting those sugars. literally affecting the auger. You can also see bubbles or cracks in the auger. So how do we inoculate TSIA? This one, we have the traditional slant surface and a stab. We need to get the stab because the stab gets the bacteria down deep where there's no oxygen, and we can observe anaerobic things like the sulfur reduction. For TSIA, we read this based on color change for the phenol red pH indicator. So that's the last question as well. So phenol red could be yellow, no change, it's orange, or pink, yellow from the acid from fermenting a sugar, pink from using the peptone that's in the broth, breaking it down to ammonia, alkaline byproduct. And the color of the slant versus the butt matters too. So in the slant, we're testing for sucrose and lactose usage, sucrose and or lactose usage. The butt is only testing for glucose usage. So yellow or pink in those areas tell you something about those sugars. We also have black precipitate formation and gas production. All right, which test is pictured here? This is one that we didn't do in lab. So this isn't the picture. If I have this one on the test in lab, this isn't the picture you'll see. You'll see the one from our actual notes that are provided in the lab part of Canvas module. It says urease test. Urease is the name of the enzyme that breaks down urea into carbon dioxide and ammonia. Ammonia is notoriously alkaline. Okay, ammonia, alkaline. Which test is this one? And also urea has phenol red in it. That's why it turns pink with the ammonia. I think it's positive for that one. This one though, often trips people up. Most commonly have this situation where we know that we might look at that with the bubbles and know this is the hydrogen peroxide test. People always want to select oxidase for this one because that is not oxidase, it's catalase. Catalase is the enzyme that catalyzes hydrogen peroxide or H2O2 into water and oxygen. The oxygen creates the bubbles. So catalase is catalyzing that reaction. Oxidase is testing for the presence of cytochrome C oxidase in the electron transport chain. The last one of all the cytochromes that move the electrons through the electron transport chain that help pump the protons out and create the gradient. Gets to the end to cytochrome C oxidase, moves that electron to oxygen to reduce it and turn it into water, right? So oxidase is cytochrome C oxidase. Let's see if there's something else I'm forgetting about that. But it's pretty commonly missed, that one. Don't confuse them for the test, okay? So what test is this one then? This one's the oxidase test. The reagent we use for the oxidase test is just oxidase reagent, okay? That's all you need to know. Probably not going to test you on that, but don't worry about what it is, okay? All right, other tests that we need to be familiar with that we didn't show on the review is malonate. It's green like citrate, but it's a broth. Both citrate and malonate are testing for whether or not you can use that for the sole carbon source. If you can, you make an alkaline product, which includes ammonia and carbonate. That'll turn it blue, because bromothiamyl blue. Deaminase, we're looking at phenylalanine deaminase. That's an enzyme that deaminates or removes the NH2 group from the amino acid phenylalanine. If you have deaminase, then you will remove that amine group. We'll add ferric chloride to the slant, and the slant will turn green if you're positive for that. If you're negative, it'll just stay that golden yellow color. MRVP is methyl red and Vogue's Pro Scour. These are technically two tests. We start with one MRVP tube. When we test for methyl red, you add methyl red and you look for it to turn red. Red is positive. Orange is negative for that one. For VP or Vogue's Pro Scour, that one you're going to add one naphthol and potassium hydroxide. That's the one that's out for 30 minutes. And you look for a red layer at the top for positive. That golden brown color is negative. MR looks for heterolactic fermentation. VP looks for mixed acid fermentation. Just different ways of fermenting. Then we have gelatin. That was a butt that we stabbed to inoculate. And then we put it in the fridge after it was done incubating and looked to see if it stayed liquid or not. If it is liquidy, then we broke down gelatin and we lost the solid structure of it. Then that liquid was made because we broke down gelatin using gelatinase enzyme. If it stayed solid, you didn't make gelatinase enzyme. All right, let's talk about bacteria in general, cells. How would you classify this organism's characteristic arrangement? I point an arrow at the specific one I'm looking at here. Because there are obviously diplo in the picture, but don't get caught up on the diplo here. Diplo is pairs. All right, so that's strepto. Strepto, strips, chains. You guys joined the Friday review, then you got to hear me talk about strippers. This is our example for strepto. What is the name and shape? I meant the name of the shape. Doesn't really matter, but of these bacterial cells. The shape of these. This is bacillus. They are rod shaped. You're only going to have bacillus or coccus. I don't have any other shapes available for you. The only arrangements that I have available are going to be Staphylo or Strepto. I do have Strepto arrangements. We didn't have them in our lab, but I have slides with Strepto, so do be prepared to see that if it is a possible answer. But I don't have anything else. Yes. Yes, exactly. All right, so for our bacterial cells, we need to know the shapes, bacilli and cocci. You know your arrangements, Staphylo, Strepto. Don't get shapes and arrangements confused. You guys aren't having a problem with that this semester, so I'm not worried about that. And remember that simple stains are a thing. If you stain your gram positive with a simple stain using methylene blue, what color will it be? Blue, right? So don't get tripped up on gram positive talk. Remember, all cells can be stained with that. It doesn't have to just be bacteria. All right, microscopes. If you observe a specimen at 100x objective lens through a 10x ocular lens, what is the total magnification of the specimen? Right, so this is 1,000. You just multiply the two together. Our ocular lenses are always 10x, and it says it on the ocular lens too if you get tripped up on the exam. We had a question like that in the last exam. You'll have the same question on this one. All right, which lens do we use with oil immersion in our lab? It may or may not be in the picture. All righty, so that one is the 100x. So know your parts of the microscope. We had the stage last time that was labeled for you guys to identify what it was and what it did. That was on the midterm. You won't have the stage this time. Okay, so know your parts and what they do for the microscope, how to use them. Remember lab safety stuff. We went over the stains and which ones you're going to need to know for that. Gram stain, acid fast stain, capsule stain, endospore stain. There's really just 4 of them. We have all those tube tests and plate tests, selective, differential, and biochemical tests that we went through. Be able to look at an identification chart like the ones you filled out on your first page for your unknown report. I get one filled out for you. And then I give you a dichotomous key to be able to use the info on the chart on the dichotomous key. Okay, well, it starts out with cocci here. Is it cocci or is it bacilli for my gram positive? This one says it's cocci, and then follow through to use catalase and whatever in order on the dichotomous key to find the name of the organism. I might also just give you a list of traits. It might say it is catalase positive, urease negative, phenylalanine positive. It was made acid in glucose, but no gas. And then I'll give you a chart. You might not necessarily have it in the table, and you might not need to use all of the information that I gave you, but then find the organism name from that too. Okay, there's 2 questions for that. One is a gram positive and one is a gram negative. Cool. All right, exam is on Thursday at normal lab time. We do still have lecture on Thursday, so it's chapter 22 lecture, and then we will go up to the lab. You guys know how it goes if we get done early with the lecture, which we probably won't, but that's okay. And we'll just go straight to the lab and start in. You guys know the rules at this point. You did pretty good and have any issues last time, so I don't anticipate being any issues this time. Any questions outside of that? You know, you can email me at any point if in your studies you get confused or if you need me to clarify anything for the course. I'm always available. No, for Thanksgiving, no class at all. Not for my class. Lord knows I need it. You want me to like explain it a little bit?
Yeah, just like I think I just.
Okay. So, starch hydrolysis, we have like a normal color plate usually. Inoculate it, and we'll add iodine to it. It turns starch, iodine turns starch, that dark purple brown color. Your organism actually uses the starch. It will, I can make this easier for you if you want.
I just didn't want to interrupt y'all's conversation.
You're okay.
Sorry.
If the organism uses the starch, it won't be there anymore, right, for the iodine to interact with, so it'll create a halo effect around the growth. We didn't have any good examples of it in lab, because I had to go for too long, like a whole week, because we had like weather or something, I don't remember what, but yeah. So usually it'll have a halo around that growth. On the exam, if I have starch, I'm not gonna have an actual bacteria with iodine on it. It'll be like, I'll say, this is the starch plate. What would it look like if it was positive?
Okay.
And then what reagent would you add? Iodine. Does that make sense?
Yes, thank you.
Yeah, no problem.
Sorry,
Yeah.
it would be a double down for me Thursday.